Okay, hello. I can see people coming into the webinar. Welcome. You're all very, very welcome indeed. It's great to have you here. To start with, very, very simple technical questions. Just please let me know that you can hear me okay and that you can see the screen here. Just drop me a note in the chat or the Q&A. Just say hello. Tell me where you're from. Very curious to see who's actually attending and where we come from. We tend to have a bit of a spread from all around the globe here, and that's always very, very nice to see. So give me a shout there in the chat. I'll just go over and have a look. And while I'm waiting for you to say hello, let me know where you're from. I will introduce the webinar for today. I'm Ben Liberton from Symcel. We're a Swedish medtech company pioneering and doing some really, really fun work in the field of biocalorimetry with our instrument, the CalScreen. And today is very special for us because we actually have a real bona fide expert to talk to you about biocalorimetry. So if you've ever heard of Symcel before, if you've ever heard of biocalorimetry, microcalorimetry, this is where we can really get in and really explore the technique and exactly what it is, what it does. So without further ado, it is my pleasure to introduce our product manager from Symcel, Dr. Malin Suurkuust, who is in the next room. We're kind of coming live to you from Stockholm. She is a biochemist from the beginning and did her PhD in biophysics. And over the course of her studies and career, has really done a lot of work with different types of microcalorimetry. And so we're going to learn a lot and find out what it's all about today. Very much looking forward to it. I'm really glad to have you all with us. If you have any questions, let us know on the go in the Q&A at the bottom. But for now, I'd really like to hand over to you, Malin. The floor is yours.
Thank you, Ben. Yes, today I will talk about biocalorimetry, and I will talk about it. There is no specific topics what you can do with calorimetry. I will give you a broad overview of the biocalorimetry field, what type of applications slightly, and what calorimetry is. And I hope I will inspire you. So I see this as an inspirational talk. So to talk about different things that you can do and you can explore. So if I don't talk exactly about what you are working with today, it doesn't mean that you can't use biocalorimetry for this, but you can get an idea of maybe I can do that and maybe I want to test that. So that's what I hope to achieve with the talk today, getting some ideas of what you can do. So we know biocalorimetry is a very powerful technique for the study of living systems. Let's see if we can also forward the presentation. I will start by talking a little about in general what calorimetry is. I will give you some ideas about types of calorimetry, some calorimeter history, how would you measure it and why would you use it? And we know that all living systems will produce heat while they are metabolizing, and this is what we are going to measure. We are measuring the heat of metabolism or the heat of life. Calorimetry is the science or technique of measuring changes in heat due to chemical reactions or physical processes. And metabolism is a lot of chemical reactions. So this, again, what we can measure. The calorimetry is a word composed of the Latin calor, meaning heat, and Greek metron, meaning measure. So we measure heat, as mentioned. And some of you may have in undergraduate studies or in school done very simple calorimetry, which is referred to as a coffee cup calorimeter, where you have a styrofoam cup or an isolated system. You put water and something that you want to react inside the water. You might stir it and then you measure a temperature change. This is a very simple calorimeter. What we are going to talk about today will be much more sophisticated and you can do a lot more, but this is just an idea. So if you have a process Like if you put a certain salt into the liquid, it will consume heat and it will get colder, so it's an endothermic process. And that's what we would measure then in a temperature decrease, for example. Evaporation is also another endothermic process, and you know it yourself. If a hot summer day, you get warm, you get sweaty, and when the sweat evaporates from a body, that has a cooling effect. So that is an endothermic process that helps your body to cool down. Then we also have a lot of exothermic processes, when heat is released or produced during a reaction and it gets warmer. Then we will measure temperature increase in this simple calorimeter. And that could be combustion reactions, oxidation reactions, a lot of different reactions, as well as metabolism that we will talk more about today when we talk about biocalorimetry. There are different types of calorimetry. You may have come across some of them. If you don't know, well, today we will talk about isothermal microcalorimetry, so you may have heard about that. Isothermal microcalorimetry isothermal means that we are measuring at constant temperature. Micro in calorimetry means that we are measuring very sensitive. We are measuring at microwatt levels or smaller amount of heat. I will later show you on a scale where that number comes in. The microcalorimeter we are going to talk about can also be referred to as a heat flow calorimeter. We are doing a passive measurement with heat that flows out of the system. There are many types of heat flow calorimeters, not only the ones we are going to talk about today, but it's a term that you can come across, and it could be biocalorimetry using that technique as well. Differential scanning calorimeter could be either a microcalorimetric technique or other calorimetric technique. But if we talk life science and biocalorimetry, differential scanning calorimetry is used to look at stability of proteins, and it's used a lot within the biopharmaceutical to look at stability of antibodies, how to stabilize them. We can see a DSC curve in the bottom here, where and this is not isothermal, you are changing the temperature constantly. Another microcalorimetric technique is isothermal titration calorimetry. It's abbreviated ITC. Then you will measure on interactions between molecules, and you will do aliquot additions, as you see up here, and you measure when two molecules bind to each other. A lot used within drug discovery to find a good binder for a receptor to have an effect for a drug. For example. So these are the microcalorimetric techniques. Then there are other types of calorimetry that you might have heard of, bomb calorimetry or combustion calorimetry. And this is actually where all our food calorific values come from. For you to take a food substance, put it in a container with excess of oxygen, and you ignite it. And then you measure how much heat is produced while you completely combust the foodstuff. So we have the calorific values on food or energy content of food, and it comes initially from bomb calorimetry. Today they don't do that, they will take it from tabulated values, but once upon a time that's how we got the values for food. Then there are more industrial calorimetry, like accelerated rate calorimetry. It's looking at safety for lithium-ion batteries, for example. How much can you heat it up before you get to a runaway reaction? Solution calorimetry, where you dissolve a sample and measure how much heat that's in the dissolution reaction. This is used a lot within the pharmaceutical industry to characterize drug molecules for solid state drugs. Reaction calorimetry, we can say that we are doing reaction calorimetry because we are looking at the reactions of life, but reaction calorimetry per definition or is more that for the process industry to optimize the reactions without getting runaway reaction. They can make additions and see how quick they do that and measure how much heat is produced at the same time. And then we have another type, what's called indirect calorimetry. It's actually not calorimetry, it's measuring oxygen consumption and relate back to if you, what do you say? For the carbon sources that you have, you can relate that back to how much energy a body is producing related to the oxygen consumption rate. So that comes back into biocalorimetry, more or less. So that was the types of calorimetry, and today we will focus on isothermal microcalorimetry as a biocalorimeter. Some history. It actually started already 240 years ago. The winter between 82 and 83, the very first calorimetric measurement was conducted by Lavoisier and Laplace. They constructed an ice calorimeter. We can see a schematic of it here. Here is the picture of the real thing. But we have a schematic. They would have a double canister, an isolated outer one, and then they have in the middle, they had ice, and then in the center of this calorimeter, they have a container where they put living animals. This shows a rat, but it was actually a guinea pig they put in. And they would measure how much heat that the animal was producing while it was living and respiring inside this chamber by measuring how much ice that was melted from its respiration. Then they also burned charcoal in this, and they could correlate the energy from the charcoal to the metabolic or the respiration of the rat, and they proved that respiration is a slow combustion process. This was very long time ago, but the first calorimetric experiment was actually a biocalorimetric experiment. Then about 75 years later, the first calorimetric measurements on the microbial system was performed in 1856, where the heat production accompanying alcoholic fermentation was measured by measuring the temperature increase in an oak fermenter. So this is a little in analogy with the what I showed, the coffee cup calorimeter from the beginning, where you have an isolated calorimeter container and you measure temperature increase. Then the industrial development or revolution came in between, and everyone was more interested in how much content fuel would have, how to start industrial processes and the energies involved in that. So it took more than 100 years before there was an increased interest in biocalorimetry again that started during the 1960s, 70s, and 80s, with much less sensitive calorimeters that we have today, but nevertheless, they were looking on samples in microbiology, looking at infections, see how they could treat that. They also used it in medicine for the study of blood cells, cancer cells, fat cells, and see how they could relate the heat production in these cells and correlate them to a diseased state. So diseased cells will generally generate much more heat. And then they will also do immune complexes to see what kind of immune reactions and how immune cells were triggered. They could put biopsies in, looking at muscle fiber bundles, and they could treat them with different things and see how the metabolites were reacted. And biocompatibility, like is there an immunological response if you put the catheter material to immune cells? Will they react? There will be a metabolic burst that could be detected by calorimetry. But yet this never started off, it didn't fly, because the calorimeters at that time, not only for the sensitivity, but also there were no multi-channel systems. So it was very difficult if you deal with live cells, you want to test them during the same cycle or the same state, and you want to do triplicates or whatever, and you might want to test different type of treatments. So the most, there were 4 channel systems, and this was not enough. So calorimetry again went into the industry, pharmaceutical industry, material science, and was a lot in that field. A little work, but it never got so far at that time. But there is a lot of work published in these early years. Then now we see the renaissance of biocalorimetry. Now we have a multi-channel system. So we can run a lot of samples in parallel. We can test different treatments. We test the cells in the same stage, and we can do screening or just testing of the systems. So why would we use calorimetry to do that? But we know if there is life, there is heat. During metabolism, we are producing heat, or every organism is producing heat. It might be very, very little heat, as we can see in the table here. We can see that human cells would produce less than a picowatt, so 10 50 watts per cell. But as long as you get many cells together, you will actually get up to a detectable range for that. So when we talk about microcalorimetry, means that we can measure in the microwatt range. So we can measure actually sub-microwatt, we can go down. So in this area, this is where microcalorimetry can measure on sample. And if we just see that one bacteria, like an E. coli, will produce about 2 picowatt. So you only need about 100 to 100,000 cells to be able to detect them in the calorimeter. And we know that bacteria replicate quite fast. So even if you put a few in from the beginning, it doesn't take very long until you have a measurable signal in the calorimeter. So if we just look at the data output that will come from a microcalorimeter, is that we will measure heat flow. I mentioned we have a heat flow calorimeter. Heat flow in microwatt, because the microcalorimeter, and we measure this in watts. Watt is equal to joules per second. So it means it's the rate. It's the rate how much heat we are producing per time unit. And if you are from the USA, you will see that you may call it calories per second instead of use per second. Use per second will directly go to watts, but if you use calories, you will say microcalories per second. You can't translate this into watts. But this heat flow that we are measuring is directly proportional to the rate or the intensity of the process or processes that we are studying. So we are measuring everything that goes on in the calorimeter. So this is a bacteria growing. We can see that we have, this is a duplicate sample. We have resolution that we see there are other things going in the growth curve. We are measuring both growth and we are measuring metabolism simultaneously. So we get a growth curve, and then if there are nutritions going out or the cells are dying off in the end. If we integrate this heat flow, we get heat, so joules or calories, if you use those units. And this integrated heat is directly proportional to the extent of the process or processes studied. This could be either you follow the CFUs. They must not agree with this curve, because we don't only have growth here, we also have the different metabolic events that takes place. It could be oxygen consumption, so extent of oxygen consumption, or production of any metabolites, if that's what you are measuring. So this is more related to the extent of the process. So our biocalorimetry solution, it's the Cal Screener. So it's a 48-channel isothermal microcalorimeter. You can run 32 parallel samples in this. Why it's not 48 is that all sensitive calorimeters need to have a reference. So we're measuring against reference, and this particular calorimeter will have 16 references to measure 32 samples. You will put your sample into our sample vials. And you can also put the small plastic insert that could be either like the plastic insert, or you can have an activated plastic insert if you want to have cells that should attach to the surface. And then you close this and you put the vials, the 48 vials or 32 ones with the sample and the references, you put them on the plate, and this is a batch measurement, so you would push all the plate in and measure all samples simultaneously in the Cal Screener instrument. That is composed of a thermostat that keeps the temperature constant. 37 degrees is the standard one, but you can also extend that both ranges to measure other type of, well, optimal growth temperatures for your organisms. And sample volumes, I don't know if I mentioned that. You can do from whatever you can fit in, 100 maybe, or even less, depends on your sample, and up to 600 microliter in the vial. The measuring principle. Now you don't need to know this to be able to measure calorimetry. This is all taken care of by the machine. You just get the output. But sometimes we get questions and it could be interesting to know how it's actually measured. So it's very, very brief overview on the measuring principle. Inside the calorimeter, it looks like this. You have your sample vial. It will be standing directly on what we call the thermopile. This is the heat detector. The heat detector is directly connected to a heat sink for this individual channel. We see they are separated between the channels, but this is also directly connected to a bigger heat sink. This heat sink and the whole calorimeter unit sits in a thermostat that keeps 37 degrees. And then you have air surrounding everything. So what we do with the physical contact onto the detector is that we are channeling all heat that is produced through the detector and out in the heat sink. So if you have a sample and that is producing heat, we call the heat P there, heat production in the sample. And that's related to the heat flow we are measuring through this heat balance equation. So we have the heat production. It's related to the measured heat flow and some kind of heat capacity term. The heat capacity term is because everything will store some heat due to a temperature difference, but for slow reactions, this will be near zero, or steady state reactions, this will not be part of the equation. It's very small compared to the whole reaction that you're measuring if it's slow reactions. Even if it's fast reactions, we let all heat flow out. So this will come out eventually as well, because we want to channel out the heat out of the system. The heat flow we are measuring, it's directly related to the temperature difference between the surrounding temperature and what temperature that is in the sample. So it's a temperature difference over the detector. And if you have a temperature difference over the detector, it will generate a voltage signal according to the Seebeck effect. So this voltage signal is what we're measuring, and we convert it into the heat flow signal that we are collecting in the software. I mentioned that all heat flow calorimeters are twin calorimeters. So this is measured against inert reference. In the inert reference, there should happen nothing, and we will take the sample minus the inert reference, and then the surrounding temperature will cancel out, and we get a much higher sensitivity of our measurements and decreased noise. So very simple how we measure it. And now I will go to the benefits of using calorimetry to study biological system. Well, we can say it's simple. You, in principle, only need to put your sample in our vials, or sample vials, close it, load it into the cal plate, load it into the cal screener, start the experiment, and just monitor what's going on. And you can fit anything that, well, you can measure on anything that you can actually fit into these vials. So whatever. And you don't need to do any sample preparation unless you want to do a specific sample preparation, and it's label free. So you're actually measuring your organisms just as they are. You don't need to label them with anything. So you measure on native organisms. It is sample independent, as we came from the last slide. Since we're measuring heat flow and heat is flowing out of the system, there is no other limitations. We can measure opaque, viscous, solid media. We can measure on body fluids with any community composition, any kind of cells or tissues, again, as long as they fit our vials. You can measure on seeds, soil, small plants. You can small animals or small parasites. It could be complex communities. You can have different, like a skin model, and you can infect it, and then you can add a treatment. So it could be very complex, the setups. You can measure in different oxygen consumptions, and you can measure on biomaterials. And here I also mean, I mentioned briefly the biocompatibility before. So that you can see if there is immunological responses by adding a implant material or a material that should be implanted if you have any response on that. But you can also test antimicrobial coating. So you can test if an antimicrobial coating will have an effect on your bacteria. Biocalorimetry is fast and it's highly sensitive. And when we talk about sensitive, I've already mentioned that calorimetry is very sensitive. We measure in sub-microwatt. So that's the detection limit of the instrument. But what does it mean for your sample? You saw that our bacteria or cells would produce very little heat as such, so we wouldn't detect one, but as long as we get up to a certain number, we can see them. And here's an example with E. coli with different inoculums. So it's a dilution series from 10 up to 7 down to 10. And we can see them. We see they have a similar profile. We can also get them because of the replication. It's not very many hours before we can still detect that we started with 10 CFUs. We will still be able to pick them up in a very short time. There is a possibility to do a standard curve from the initial inoculum size to time to, in this case, it's time to peak. So time to peak is plotted against the inoculum. We can also, we don't need to go to the peak, but you can also take where we have time to detection instead, where you see that the exponential is starting or it comes over a certain threshold, and you can see time to detection. Now we can't do that here because they are too much, so we would never, well, they are already present. We have already detected them already from the beginning. But if you have diluted solutions, you can also use when the exponentials start. And it's fast. We can see we can detect few bacteria in a very, very fast compared to other methods as well. It is direct and continuous. We are measuring continuous in our sample since from the time we put it in until we take it out, we will measure continuously what's going on. So we will see what happens in our sample when it happens. You don't need to do any sampling, you get kinetic information, and it's not an endpoint measurement. You will see it throughout. Now the technique is non-destructive, so you can analyze it afterwards as well if you want to have another type of endpoint measurements that is possible. I just found 2 examples in the literature here where the curve here that's have a lot of events here and goes up and goes down. This is the heat flow data. So we're looking at growth and metabolism. There are several events going on here. The red dots here is the OD and the other black one here is the integrated values. We can see that the integrated values that I mentioned, it's like the extent of the process. It correlates quite well with the CFUs, with the OD increase here. So we can say that this correlates with that. But when we integrate the data, we lose a lot of information because there is a lot of things going on here. Could you be used to identify your organism, or if you have other parallel measurements going on, you can see what is going on, which metabolites, what is it doing during this time. Another example here with the CFU increase in red again, we come to stationary phase. You may think this is ready, but if you measure in microcalorimetry, well, there's other things going on. What is going on? Calorimetry will see there is something going on, and then you need to find out what's going on. If you have another method you're measuring, you might not see this resolution on your data. So what we say that heat doesn't come in different colors. So it's a very general technique, and it's non-destructive, as I mentioned, but it's an ideal detector to find out about unknown phenomena. You don't need to define what you're going to look at. Put your sample in, see if there is something going on. Then you might do other analysis afterwards because you want to find out what was going on. But if you, for example, only measure oxygen consumption, you may never see the other effects that you would like to see. So you can detect very complex events. It's not sure that we can explain them, but you can see that something is going on. Again, here is data from sometimes in the 90s, I think, which republished later on, where you have the black one, E. coli growth. It drops very suddenly. There were parallel measurements done by measuring the oxygen, and we can see at the time that the heat flow, the metabolic activity goes down. We also see that the oxygen levels goes down. So what we can see here, the growth starts again. And this is when it changed from respiration to fermentation. The green curve here, it's not on the scale, but this is OD measurements that goes on. So we can see there is a continuous growth of the data. So calorimetry is a unique and information-rich technique. Now I just show very few examples here. There could be a lot more that you can do, but like very schematically, there are some things that you can very easily analyze from. We talked about time to detection. It could be when it rises a certain number over the baseline. And I put X here. It will depend on how your growth curve is, if it's steep or if it's very shallow, where you see you can take your detection point, or if you have a background, a cell background or something, that could be a little different where you put your detection limit. Time to peak you can use otherwise. Peak height can be interesting, and as you saw on some of the other graphs, you can have several peaks, so you can quantify for several different peaks. You can analyze the area under the curve, so the total metabolic heat that is produced for a certain organism. But then you can also fit data by, in this case, they have integrated. You can fit different growth models. Usually the growth is best fitted from the beginning because then other metabolic effects will come. But it's possible to use different growth models to evaluate lag phase, growth rates, and what do you call it? Duplication number. No, sorry, I maybe not the right word, but how quickly the bacteria replicates. It's also possible to do modeling to see the different type of, if it's respiration or fermentation, you can deconvolute data. And this is just a few examples. As I mentioned, there's a lot of work going on how to interpret biocalorimetric data to find out more information, because there is a lot of information hidden in this data. But you don't need to put numbers on them. You can do both qualitative and quantitative analysis of your data. If I now split it into microorganism and cells, so microorganisms, you will look at metabolism and growth. You can put how quick they grow and how many they can do. We can study the biofilm formation or biofilm treatment. We can detect infections. We can detect contamination in foodstuff, for example. Bacteria, as you will see, will have different type of shapes, different species or different type of bacteria. So you can do identification. You can test antibiotic susceptibility on your bacteria, and fingerprinting, or because they have their specific signature or profile, you can actually look at, and if you change something in the composition, do you change anything on your organism? And then if we look at cells, they don't grow or proliferate that fast. So usually when you're measuring on cells, you will get metabolic levels of your in your measurements instead. So even if you can't analyze this by number, you can do them comparative. Like I mentioned, if there are diseased cells, how severe or what do they do depending on how much heat they put out. You can rank cells, or if you're going to produce a cell line, which is the optimal composition, and you can rank different compositions of the media to see they get best activity, if that's what you're looking for. And you can look at the effect of treatments. So now I just listed a lot of different applications you can do. So I have examples for microorganisms, for cells, tissues, looking at small animals, plants or seed, within the medical field, biopharma field, biotechnology, food and beverages like probiotics, looking at medical devices, but more the device effect, like coatings or biocompatibility. In drug development, also in ecology and other areas. And now you see there are gaps, there are holes here. This is just what I have found in the literature that biocalorimetry has been used for. I may not have found everything, may not been published, or so there. If there are gaps, it doesn't mean that it is not possible. It's just that I have not seen examples of it. So I will give you a few examples from this. We can look at for the microorganism, how you can identify your microorganism. So here is different bacteria. They are grown in the same broth or the same media, and you can see that the different bacteria will have completely different profiles. So this is a way to identify the bacteria, what type of species are there. We can also see that depending on the media composition, the same bacteria, like here in this example, the E. coli, will grow on a completely different profile in different type of media. So this can also be used for identification. If you have some bacteria that grows better in other, you can use specific media to look at how they grow in that media. Then another example here is to look at culture optimization. So if you want to have an optimal culture of whatever you are growing, a probiotic, for example, you may have, like in the standard media, this is the gray curve here, you have that activity, and then you put additives. This is different carbon sources, both carbon source and the amount of carbon you put into it. We can see it doesn't affect the initial growth, but we can see it affects the metabolic activity, maybe after growth, that they get more, if they have more nutrition or more carbon in this case, they can grow with a high, or they metabolize with a higher activity. The gray one is without this additional carbon. If you add glucose, we can see it doesn't matter which concentration of glucose, but you increase the activity of the microorganism very, very much just by this addition. For the pyruvate addition, we can see the lower concentration doesn't affect the growth or the activity very much, but you need to add more to be able to get that effect on the activity of the bacteria. Then another example for drug development, you could use it to look at antibiotics, antifungal drug, or phages, as in this case. You can test different phages on different bacteria. In this case, there was a resistant strain with increasing amount of phages added, but you see no effect whatsoever on the growth or delay in the growth of this resistant strain. If you have a susceptible strain, you can see that you're actually moving with increasing concentration of the phage in this case, and actually the highest concentration here is completely inhibiting the growth of this strain. So we can also see how much needs to be added and what type of effect does it have. We can also say that it both pushes this to the future or longer in time to be able to be detected, but it also changes the shape, so it could affect different parts of the organism. And then there is an example with cells or tissues. This is a cancer microtissue that is put into the calorimeter. We can now see there's different numbers of these microtissues put into the calorimeter. We can now see that cells are producing less heat in total compared to the microorganisms. So if you have hepatocytes in this case, or the cancer cells, they will produce less heat than a microorganism when they grow. They were also seen by putting in increasing amounts of these tissues. These tissues are composed about 1,000 cells in each tissue. You can see the more heat produced for the higher number of tissues that puts into it. Here has also been growth model fitted, and you can see how quickly the proliferation of these cells are. You can calculate from this. After the measurement, they were taken out and checked in microscopy, and they could also see that the tissues has grown afterwards. So this is what we mean with the non-destructive. You can take it out and do further analysis afterwards. We can also see that the timeline of this is much longer. I forgot to mention it when we looked at the microorganism, but that's hours, and now we're talking days here, so that would be different. And then this is the last example. This is where parasites put into the calorimeter. So here is 5 schistosoma worms put in, and we can follow the metabolic heat over time. What also been seen here is that it's excessive noise, and the noise actually reflects the motor activity of these worms inside the calorimeter. So it could be the overall metabolic activity as well as the motor activity of these parasites. So here it was 5 put in. In this case, it was only 1, but still you can see the motor activity on one single parasite in the calorimeter. And then when signal the noise or with the motor activity and the metabolic goes down, then the worm is dead in the end. So that was a few examples. If you want to look at this or fill the holes or try what others have tried, find inspiration. There is a lot that could be done with microcalorimetry and biocalorimetry to look at that. Please. So biocalorimetry going forward. Well, we don't see that the full potential of biocalorimetry is still being explored. We have not met its full potential yet, and that's so exciting. And there are great opportunities for qualitative investigation with, like I say, just comparative, look at things, compare things, and see if treatments. But we can see that the future of biocalorimetry will depend less on instrument improvement because we have good and sensitive instruments now, but it's more, we can always improve, of course, but we see that more and more will go to interpretation of the data from these complex systems. How much more can we understand of what's going on in our living cells? And we see there is a continuously increased knowledge of the processes in this complex system. And with this, it also opens up the possibility to interpret calorimetric data, not only in phenomenological terms, but also quantitatively. We see that in the future, we will be able to put more numbers on what we are measuring. So to conclude, so it was almost a quarter of a millennium since the very first biocalorimetry experiments was conducted. And now we have evolved into the century of biology, as stated by Craig Venter. And there is an ever increasing interest in an understanding of how biocalorimetry is or can be used. So there's a lot more. We see there is a lot of ongoing applications, is very exciting, but also going forward, we will see much more interesting information coming out using calorimetry as a technique. And I just want to conclude like some benefits of calorimetry. We say it's universal. Heat is a universal detector because all processes generate heat. We don't need to define anything else. Put it in, look at it, and we do it at an extreme sensitivity. It's one method. You don't need to define what type of response do you have. You don't need to say, I want to look at the metabolism. I don't want to look at, or I don't need to look at the oxygen. We are looking at the overall response of a metabolizing system. You can get metabolic activity and growth from the same experiment. It's direct and continuous information, so you get a completely kinetic profile of your process under study. And now we have a multi-channel instrument for increased throughput. I really hope you got inspired by this, find some use of the instrument, and I hope you will follow us with a journey to take biocalorimetry to the next level. Thank you.
Thank you very much, Malin, and thank you everyone for attending. before we get into the questions, I know some of you said you had to dash off. So if you have any questions or you think about follow-up, or you want to engage with us more, please just go to the website. You can email Malin directly there. You can see her email address on screen. So thank you again, and thank you to you if you have to dash off. But let's get into some questions. I can see people coming into the chat already. If you want to send in the chat, there's the Q&A. You can reply to this email. We're going to be monitoring them all. Start with Malin. We had a few questions prior to the webinar even. So we had a question about what exactly is being measured if you were to compare biofilm and supernatant. How could you differentiate the two?
Yeah, it's not, what do you say, straight on simple to do that, but there are different ways. So it depends on your experiment and how you set it up. So that's something we would discuss, how to set up that particular experiment, how to do that. But there was recently a paper by the group of Björnsholt that published how to do also PCA analysis or principal component analysis to see how you can evaluate this difference. So we will refer you to our homepage, or we'll send some literature to be able to look at more in detail, or we take the discussion for that particular system that you would like to look at.
Fantastic, yeah. And we will send more information on that to you. Another question from the same person about imaging. You covered this a little bit, Malin, but maybe it bears repeating. After you've done biofilm experiments, you want to image them. How do we kind of handle these kinds of requests and experiments at the moment?
Yes, we do have, like, the inserts we are having, they are microscopic ready, so you can actually take them out after the experiment and look at them in a microscope. The same paper I was referring to has done the same. There are other groups that done that. Those papers are listed on our webpage as well. So yes, it is possible to look at them microscopy or image them after the measurement in the cow screener.
Great. What about anaerobic bacteria? Can we do those? How do we seal the wells?
Yes, we can do anaerobes. Well, you need to seal them in an anaerobic chamber. When you load your sample, you need to handle them anaerobically. But once we seal them in our system, they are sealed, so there will be no oxygen coming into. So if you seal them under right conditions, we will be able to measure them in the machine later on. Yes.
Fantastic. We have a volume question now, Malin. So working with polymer encapsulating E. coli using 50 microliters of sample, is this something that can be measured?
50 microliter of sample? Yeah, as long as you can use as little of sample as you want to do it, as long as we get enough heat signal. And that's if you have them encapsulated, would you now? I don't know if you would do dilution of them, or you just want to put the encapsulated thing into something else. They need to be growing over a certain amount or have a metabolism over a certain amount. If they have that, you can use a small sample as you want to do. Usually you will dilute like your E. coli in media, and then you will add more media, but you don't need to add very much inoculum into them. So.
Great. Yeah, I hope that answers your question, because you mentioned there in the question about accuracy and reproducibility. So I guess the response is kind of, it depends, but if there's anything unclear, please ask for a clarification in the chat or the Q&A. Another question here about the overlay graphs that you showed, Malin, with the OD versus heat, and how they were actually done.
Now, usually when they do this OD, well, you would like to do them under the same conditions. So if you want to do OD over time, you can do a parallel incubation in these vials outside, and you sample from the parallel measurements, maybe in an incubator over time. Then you will have exactly the same conditions. Now, I can't really answer how these publications were done, but this is very lot of times they will do sampling from the same conditions that you will have in the instrument, because there will be different surface areas and there could be other things affecting. Yeah.
Great, thank you. Again, any clarifications if you need them, or any follow-up questions, please feel free to drop them in. Malin, does the location of whatever you're measuring matter to what heat you measure? Like if you had like a pellicle biofilm floating on the top compared to a surface attached one, would you see differences there?
All in our system, it's sort of a twofold question here or answer. We have a heat flow calorimeter. We channel all heat going down to the detector, as mentioned, and so all heat that we're producing will get out. We will measure all heat that is produced. If there is on the top or on the bottom, it could just affect the time until. So you can get maybe a little skewed curve depending on the heat needs to transfer from the top down to the bottom. Could be, but doesn't say it has to be. So it depends a little on what type of medium and how quick it goes down. But all heat that is produced will get out.
Yeah, this was one of the biggest things that when I discovered this, because you assume with methods like this, I think that everything is on the surface. You know, there's some detector on the surface, and then a lot of people, when you realize it doesn't matter, it can be anywhere, it kind of changes the way you think about it, and it opens up a lot of kind of new experimental ideas. Fantastic. Let's have a look. Any more questions? Keep them coming if you have them. Oh, technical question from the lab. Can you load the plates with a multi-channel pipette?
Yes, it's possible. It needs to be one of those adjustable pipettes, but then you should be able to load with those pipettes as well. Yeah. So a 96 well adjustable pipette could be used for our plates as well.
Great. Okay, also, so you measure anything that produces heat, so that's the metabolic activity of the organism, but you know, do other processes, degradation, other reactions contribute to the signal, and how does that impact your interpretation?
Yes, well, everything that produces heat, as you said, will be detected there. Usually when you do metabolism, you can have a ground reaction. Maybe there could be most systems we're looking at, it's only the bacteria that produce heat. But if you would put in something in your media that is not stable at the temperature test at, then of course that could give rise to heat as well. Usually you would do blanks and see that the media is not contributing anything. But if you, for example, would run with, what's it called? Sorry, but some where you have a lot of other biological material in, or it reacts with each other, then of course that heat will also come in, but you would do blank measurements for that. For the most of the media and buffers and everything we are using is not producing any heat that contributes anything. Otherwise you would do blank measurement if that would be the case. Yeah. This is such a low temperature for very many other chemical and material reactions. So it's too low heat to detect anything that contributes to the heat that we are measuring.
Great. Are there any limitations when it comes to media and, you know, what you can actually put into the vials?
No, as long as you can put it in, you will. There could be, as I mentioned, if there is a reaction in the media, then you need to see that what this reaction is, so you have your blank measurement, if that would be the case. So yeah, that's what's the word I didn't find. Serum. If you run serum, that could be things that goes on in that, but usually that will be like a standardized. So you do blank measurement and you see what that is, and it won't contribute to the other heat that you're measuring. So you can still do it. I will conclude that you can still do it, but you just need to check off how much that heat is.
I see. So biocalorimetry, microcalorimetry, it's a novel technique for a lot of people, but it's been around for a long time. So, you know, what's taken so long for it to be taken up by different people, and what do you think the challenges are, and maybe contrast that against the benefits you see for different fields picking up microcalorimetry now?
You know, also why it's taken so long is that the knowledge, we don't learn this in school. Well, when we learn calorimetry, you have that coffee cup calorimeter. That doesn't inspire you to start working with calorimetry. And when you hear, often when I talk to people, when they hear calorimetry, they relate that back to that, and they have not even thought about it should be something else. And so I think it's not part of education. And when in the 70s and 80s, when they did a lot of that, we didn't have the multi-channel instruments. So it ceased, even if you saw there was a lot of potential. So it's both the development that have been going on over the years, and sensitivity and multi-channel things that comes today that will open up new doors, I think. But still, we shouldn't be afraid. As I said, you don't need to put numbers on, you can do comparative. So if it's a physical technique, but you can still use it, but it's, if you don't know it, I think that has been a limitation because the knowledge has been so small. But I see great potential, and the more that we talk about it, we also see that, oh, I can measure this, they get ideas, and the more that uses it, we get the spread of the word that biocalorimetry is very useful for very many applications. And if I would say, where do we see the most benefits going forward? I think we can see it. I can't say there and there. We can just see that starting to use it, we will learn so much more about these systems. So, yeah.
Great. We're getting some more questions coming in, and we're rapidly approaching the time now. So perhaps we can try rapid fire, cover as many questions as we can now, Malin. But if we don't get to you, we'll email you the answers to the questions. So what's the stabilization time before you can begin measurements?
You need to equilibrate the samples before they get into the calorimeter. And that would be, it's about half an hour to get the batch loading of the vials into the calorimeter, into measuring position. And then the loading disturbance need to go away. And that depends on, yeah, the more heat you're measuring from your system, the shorter the time. But I would say it could be from 20 minutes up to 40 minutes before, depending on how much heat that you are looking at and what type of, it depends on the time constant of the system that you put in and how much heat that you're actually going to measure. For low, if you have a very low heat producing, then you would maybe want to wait even longer. But if you have bacteria growing, those bacteria will grow over your disturbance very quickly. So you will see a signal that's much higher than the disturbance. So, but sometime in that time frame, so within an hour from the loading, from you load the sample into the vials until you have equilibrated and can trust the signal in the calorimeter.
What about calibration? What do we need to calibrate the instrument?
There is no need. It's factory calibrated. It's calibrated before you get it. So just to make sure that the heat that's produced in the vial correlates to what we're actually measuring. This calibration is done at the factory, and usually you don't need to change that. There is, if you would have a service contract, there will be calibration, like checking the calibration that it's still valid. But this system age very, very slowly. So, but you could do chemical calibration. There are chemical calibration substances listed in the literature, like hydrolysis of triacetine, or there are other substances you can use to follow very slow reactions to see the level, and not to calibrate it, but just to check if you would like to check. Normally that's shouldn't be necessary. So.
Last question. We've got a minute. Okay, so are the plastic inserts reusable?
I think they could be if you wash them, but that depends on what's grow on them. If they will attach to, and yeah, you get the biofilm formation on them. I don't know if you would like to reuse them, but so we sell them as disposables. Of course, you can try to wash them. I'm not come across that, so I shouldn't speculate in that, but they are plastic materials, so if they're washable, they might be. Yeah.
It's possible to do the experiments in the titanium vials as well, which are definitely reusable.
Yeah. Yes,
it's
you don't need to use the inserts either, depending on, it's easier because you can just throw them away generally, but if you have bigger samples, well, the volume will go down if you have the inserts. So if you have bigger samples, then you may also don't use the insert. Yeah.
Great. Well, there we go. We've hit the hour. Thank you so much for great engagement and lots of questions. Thank you, Malin, for teaching us so much this afternoon. And without further ado, I'd like to, wish you well for the rest of your day. Please get in touch with us if you have any more questions, and you'll be able to have access to the recording of this, so you can go over it again at your leisure. We'll email that to you. So thank you very much. And until next time, see you later.
Yeah, thank you.